It's such a pleasure to be here, and I'm so grateful that Alex and Ben invited me to moderate this session. And when they did, they gave me the choice of a number of sessions, and I picked diagnosis on purpose. And one of the biggest reasons is not just because diagnosis is really at the very beginning, at the heart of everything we do in public health, so you need the very, very best, accurate, informative diagnosis to start the health journey, but also because I also lead the National Strategy for Vector-Borne Diseases at CDC, which is a 17-agency strategy to try to respond to vector-borne diseases. And we have 5 goals, and they are understand is number 1, number 2 is detect and diagnose, number 3 is prevent, number 4 is treat, and number 5 is implement programs. And in May, we had a group of about 100 people, some of whom are in the room here, sit down and help us prioritize the 19 strategies in the national strategy. And the top 2 across those 100 people by far were better diagnostics and better guidelines for diagnostics. Those were the 2 top out of the 19 by far across researchers, clinicians, patients, public health workers, everyone. So I'm really happy to be here and to hear about all the great work that these 4 amazing professionals have been doing in the area of IACI diagnosis. So to begin, we have Dr. DuPont, who is from the J. Craig Venter Institute. We have Dr. Tall, who's from the Massachusetts Institute of Technology, Dr. Breitschwart, who is from North Carolina State University, and Dr. Sobieszczyk, who is from the Columbia University Irving Medical Center. And so we'll start with Dr. DuPont.
All right. Thank you very much. So I'm going to talk about something that we're unimaginatively calling the tissue profiling pipeline for infection-associated chronic conditions. And this is something that we've been cobbling together with NIH funding, Cohen Foundation funding from the PolyBio Foundation. And this is a very crude overview of this pipeline. Essentially, we take a tissue or biospecimen. A lot of the work that we're doing is actually working directly with surgeons to capture medical waste. So one of the first things we did was start to collect clots from people who had strokes. These are normally chucked out. And one of the first things we did was metagenomic profiling of these clots to find out that they were significantly enriched with microbes either from the gut microbiota or the oral microbiota. And we've started to put on more and more technologies onto this pipeline. So my background is in oceanography, but what I did during my oceanography degree was next-gen sequencing and synthetic biology. So I became a technologist. That is, I like to put together multiple technologies to address difficult problems. And so we take this tissue sample and we essentially try to create a complete catalog of all of the cells in that sample and everything that they're doing. So this includes all of the host cells, the specific types of host cells, what their transcriptional profiles look like. This is a way if we can tell, so say in a skin biopsy, is there an immune infiltrate into that skin biopsy? Are the epithelial cells expressing themselves differently? Further, we use spatial transcriptomics, and this is led by Dr. Ferrer, who's in the audience, to look at how are these host cells interacting with each other from a spatial organization perspective. And then finally, the thing I'm most familiar with myself is unbiased metagenomics and metatranscriptomic sequencing. And this all feeds into a giant matrix of data sets. And this is where we use machine learning and non-generative AI, so learning AI, not generative AI like ChatGPT, as well as differential network analysis to identify not just features that are diagnostic of a condition or not a condition, but rather interactions between features. And we believe this captures more subtlety and get us closer to mechanisms. And our primary goal with this pipeline, we're not going to run this pipeline on every subject out there. It's the rough cost of this is close to $30,000 each. This is not feasible. But rather to use small subsets of data to identify subsets of 3 to 5 proteins, genes, transcripts that can be then assayed for cheap, accessible methods, something in the range of $10 to $30. And this becomes accessible both to personal people as well as insurance cutouts. So I wanted to step back and talk about the human microbiome and virome a bit because I've gotten a lot of questions about this over the years. And why do we do both metagenomic and metatranscriptomic sequencing? So the human microbiome, everywhere in your body, you have a different community of microbes. This is dictated by both your immune system as well as the physical conditions. Obviously, your large intestine is a different chemical and nutritive environment than your skin. But the immune environment in both of those is different as well. And so our communities of microbes are very vital to our health. When in a healthy condition, they can prevent pathogens from taking shape. You have about 1-to-1 human cells to microbiome cells. This is bacterial or fungal. We're ignoring the virome at this case. It's roughly about 5% of your human body mass. Though if you get Ebola, then it roughly spikes up to about 15% to tell you how bad Ebola is. It's also highly diverse. It's influenced by diet, your host genetics, a variety of things. And so we know that there's really good biography across the body, and this is from the Human Microbiome Project, the phase 2. I wouldn't worry about looking at the colors here, but each color associates with a different body site. And so like the blue down there on the lower right, that's the oral nasal kind of complex, the oral nasal pharyngeal complex. Now, in this study, what they did is they mapped the reads from the metagenomic or metatranscriptomic sequencing to every known microbial genome. Now, there's a problem with that, in that every time we do what I'm going to show on the next slide, we discover entirely new taxa of microbes. And this continues to happen despite this discovery cycle. It's been going on since about 2006. Another issue that I'll touch on very shortly is that many of the sites that we want to talk about in infection-associated chronic conditions are hyper-enriched in human DNA, which means that you can't sequence either the viruses or the bacteria in those samples. So we've developed pipelines. I wouldn't worry about looking at this unless you're interested in the code, which is publicly available at GitHub. We try to make all of our data publicly available, as well as all the methods. But this allows us to take these individual reads. When you sequence a microbiome, you have a bunch of puzzles who have been scattered into pieces, thrown into a big box and mixed together. And we have developed computational methods to take that and reconstruct it. So in these constellation plots in front of you, on the left are all of the viral genomes that we assembled from this, and this is why we do metatranscriptomic sequencing, so we can assemble RNA viruses. On the right are all the bacterial genomes. Now this is from an oral plaque swab. That's mostly microbiome. So if we think about blood, nasal swabs, skin punches, meningitis, cerebral spinal fluid, all of these samples are greater than 99% human DNA. So we've coupled up with an industry partner called Jump Code Genomics, and we're developing methods specifically for Lyme disease and acute Lyme disease, profiling of pathogens, viruses and bacteria that might be in blood. In this case, we're using over 1 million guide RNAs and CRISPR-Cas technology to degrade the host transcripts out of the sample, thereby enriching transcripts for the bacteria and viruses that are in those samples. And we are going to have a lot of things coming out in this space soon, so watch this. And then thank you to the various funders, etc.
Thanks so much, Dr. DuPont. All right, and next we're going to hear from Dr. Tall. I would love to hear an update from you on how we might develop a diagnostic for chronic Lyme.
Thank you so much. So I asked Dolly to draw me a Rubik's Cube that says infection-associated chronic illness. And this is what came out. And at first I chuckled, and then I said, oh my God, this is perfect, actually, because I actually think this reflects the current state of our understanding of infection-associated chronic illnesses. We almost start to see how the pieces are going to fit together, but we currently have some missing pieces and some pieces that we think are right, but they're wrong. And so I actually think it's perfect. And I have 3 questions on here that are kind of going to guide what I want to talk to you guys about, but these are, I think, some of the biggest questions that we're trying to really look at, which is why doesn't everybody bounce back? And why do these infection-associated chronic illnesses skew female? And how do we distinguish very early in the immune response to the infection responses that look like they're protective from responses that are catastrophic, And if catastrophic, what flavor of catastrophic? So we have a clinical study going on at MIT, and this is the largest clinical study that MIT has ever undertaken. So I'm very grateful that they let me just come in and start taking over the Translational Research Study Center. And what you see here is that we have 4 main cohorts, but this was built to expand. And our cohorts are healthy volunteers who are actually all recovered. So they're all recovered from COVID, and a subset of them are also recovered from Lyme. And they're getting compared to people who are within 4 weeks of their tick bite and diagnosed with Lyme, or people who are over a year out, not recovered, or people who are over a year past their COVID infection, not recovered. And so the way that we've set this up, we have just acknowledged that we have no idea what's going on, and we really want to understand it and characterize it. And so the theme of Maestro is essentially to leave no stone unturned. And so you can see the slew of different neurological and neurocognitive testing that we're doing, where we're comparing commercially available devices that could be used in the clinic immediately to standardly used clinical methods, as well as skin barrier testing, hypermobility testing, different physical assessments. And if you look at our paired biological sampling, you'll see that basically we're asking people to give us anything that they're willing to give us in terms of biological samples that we're trying to collect. And if you hear of anybody who gets Lyme, we are actively recruiting, and our acute Lyme cohort has been the hardest to fill. So as an immunologist, when I look at this, I'm thinking about how the immune system is trying to kill the pathogen that has invaded or prevent it from replicating, and in doing so it can cause disease. And the pathogen can also cause disease, but I feel like our biggest question that we have to ask is, how do we distinguish, in the event that we see illness, who is causing the disease? Is it the pathogen or is it the host? And there's been this long-standing dichotomy between, do you need steroids or do you need antibiotics? And I think it's much, much more nuanced than that, and that's what I want to understand. So the way we've been going about this currently is you get a blood test and we do diagnostic serology, but this is very low resolution. So currently what we're doing is we're looking for one type of antibody, and we're looking for a very general, inclusive kind of category of that, and we're just counting it, essentially. So that looks like this. What my lab is working on developing is something that we're calling predictive diagnostics, where what we do is we're using the pathogen itself as a bait, and then we're looking at all of the antibodies that are bound to it, and we're looking at it at the resolution of which types of antibodies they are. I'm going to explain why that is. So what you see when you look at this is that different types of antibodies bind to and recruit different types of immune cells. And these immune cells have different functions. And so these antibodies aren't just biomarkers, they're actually effector proteins that drive different kinds of inflammation. And so if we move beyond thinking about the immune system as inflammatory or anti-inflammatory, but start asking about flavors of inflammation, we can actually get much more therapeutically informative information here. I'm just going to very quickly mention that John Alcott's work has done some really important work highlighting also that there's some important sex differences in Lyme diagnosis, and we're also looking at that as well. So what I want to show you here is that we have many kinds of antibodies, and we have 5 major isotypes, and of them we have different subtypes. And what you're seeing with the blue circle is IgG, and currently that's all we're looking at with this diagnostic serology, or if you're very early in infection, maybe you will also be examined for IgM against Borrelia. But I'm highlighting for you here just the example of how different the response that's going to be triggered by IgE versus IgG, and that's going to become important. And IgE is this type of allergic type of antibody. So here I'm just showing you how this works. We have the Borrelia itself. We use it to essentially as a magnet to pull out all of the different antibodies that bind to it, and then we can look at all of the antibodies that are bound and look at what types there were. And this is what these illness trajectory maps look like, but here I'm just showing you one time point. This is samples that we got from Liz Horn from the Lyme Biobank. These are all people who are over a year past their Lyme infection, never recovered. And the important thing that I want to pull out for you from this data is that a third of them were making very high levels of IgE against Borrelia, this allergic type of antibody I had mentioned. Which brings up a very bizarre question that I didn't think I was going to be asking when I started this research, and that is, can you become allergic to a bacteria? And what would that mean, and what would that look like? And I think that helps us start to think about, when we saw a third of people with that phenotype, is that already one way to start breaking people into therapeutically meaningful groups? And so what I'm showing you here is that we do this profiling, and then we're looking at it to try to understand, are you making, very early in the infection, are you making a protective immune response where you're going to get antibiotics, you're going to recover, you're never going to think about this again, versus are you making a catastrophic immune response? Are you going the wrong way down a one-way road, and maybe we could stop you before you get really too far down this? And when I'm talking about catastrophic immune responses and flavors of catastrophic immune responses, I'm showing you here what I mean of, some examples of different flavors of catastrophic responses. Now, we're also building a mouse model in my lab, so this is the other main focus of my lab, is to just try to model this and understand the mechanistics of this. Of course, we can't recapitulate all the features of the human disease, but we're trying with this. We have some papers already out, and some more coming soon. So I just want to thank my team, and we have funding from Emily and Malcolm Fairbairn to support this work. Thank you, and at R01 from the NIH.
Thanks so much, Mickey. And now on to Dr. Breitschwart, who's going to talk about neurobartonellosis as a cause of IACI.
Thank you, Susan. When I was first invited to give this presentation, I would ask my question, what can I do that's useful in 8 minutes or less? And I thought, well, how about a case that might actually illustrate this? In the context of disclosure, I do have to say that I am a founder, shareholder, and still the chief scientific officer for Galaxy Diagnostics. So on to our case. When I was first introduced to this individual in this picture, he was 8 years old. His mother had contacted me by email, and she sent this picture of the cat scratch by a feral cat that occurred when the child was 2 years of age. So question number one is, does this cat develop cat scratch disease? And no fever, no lymphadenopathy, and no symptoms. So I think we can say, no, he got scratched by a cat and he didn't develop cat scratch disease. But question number two would be to you, and particularly the physicians, does this child develop neurobartonellosis? And you'll see my comment was to be determined. Sometimes in life you can just be unlucky. So when the child was no symptoms for 2 years, when the child is 4 years old, he wakes up one morning and has this rash. Mother takes a picture of the rash, and that's the end of the story until 1 week later. And so question at this point in time, when the child has his first seizure, is does the child now have cat scratch disease encephalopathy, which is well characterized, but usually occurs in association with the acute onset illness or shortly thereafter, or question number two, does the child now have neurobartonellosis? So this slide basically shows us that from 2018 through 2021, this child is hospitalized multiple times for status epilepticus, cluster seizures, focal seizures. The seizures become progressive. Initially started on one anticonvulsant, and then you'll see down in January 2021, was started on 3 anticonvulsants because the seizures were refractory, okay? And, oops. So the workups at that point, which I didn't go into great detail, were extremely extensive with EEGs, serial EEGs, serial MRIs, a tremendous amount of commercial diagnostic testing to see if this child actually has Lyme disease, cat scratch disease, or some other disease. And the bottom line was all that testing was negative. And so it was at that point that the child had right-sided hemispheric disease with a differential diagnosis of Rasmussen's encephalitis, which I had never heard of as a veterinary internist, but it is a rare disease, fortunately, and it's actually treated by taking out the right hemisphere surgically to try to control the seizure disorder. So at that point, we got involved, and the first testing, and this again is where I think a single case can demonstrate both medical complexity of chronic illness, but also the microbiological complexity of chronic illness. And you'll see that we did testing in the lab with enrichment culture, PCR, 3 different samples collected over a 7-day period, serology against 5 different Bartonella species antigens, actually including the one you'll see in a minute he was infected with. Everything was negative. 6 months later, he has a brain biopsy at Sick Kids in Toronto, and the parents, with multiple emails and various other things that we had to go through, made sure that the brain biopsy came to us. It was put into 2 media, the Bartonella alpha proteobacteria growth media that we developed at NC State, and then a Brugge media, which is cell culture media that we use in our cell culture studies. And in both culture media, in multiple samples over a 21-day period, we could document the presence of Bartonella, and we could actually amplify it and sequence it and say it was Bartonella henselae in the San Antonio 2 strain, which is a strain we use to test in our IFA testing. And so that was, okay, now we figured out this child has Bartonella, it's in his brain, it's 6 years later, and the fact that we couldn't detect it by direct PCR from the tissues and we can detect it from the cultures, that it is possibly still viable within the brain for a long period of time. So thanks to the Cohen Foundation, we initially looked at Bartonella digital PCR and then went on to look at Babesia and Borrelia droplet digital PCR. And so in the process of testing a whole bunch of samples that we were just serially testing, this child comes up positive in our digital PCR for Babesia. And what you can see on this slide is on multiple occasions, and these results are based on DNA sequencing of what's called the energetic spacer 1, energetic spacer 2, and 18S gene of the Babesias. So we've got 3 different areas that match up that we can get and say this is definitively a Babesia divergens MO1-like strain, which has been reported by infectious disease physicians in the United States. And as you'll see, Babesia odocolii, both of these are ruminant Babesias, and as a veterinarian, the fact that we're finding, which other labs have clearly done around the world, ruminant Babesias in a 2-year-old child was not what I expected. And I just wanted to end with this. So our most recent publication is on Babesia odocolii and Bartonella species co-infections in the Americas. It's just a small case series of 7 individuals. But what is important to me as an internist is that the child in Oklahoma that was 2 years old that is published in that paper, and so that's her pictures, had a lesion that was very close to the child in Canada that, you know, so we got one I think was in June, the other one was in September, and similar looking lesion, and both infected with Babesia odocolii. The only difference is we had a co-infection in the child in Canada. So I think there's a lot to learn in regard to what's going on with Babesia right now, and we've got a lot more data in the lab that will be coming out shortly. So my final slide is cat scratch disease, what's in a name? And I think physicians have to stop using the term cat scratch disease. I think we need to go to Bartonellosis. We need to realize that it can persist for very long periods in the blood and apparently in the brain, if you believe what I've just showed you. I have mentioned several times, commercially and otherwise, we were never able to find an antibody response in this child. Presumably, he was infected by that feral cat with a right facial cat scratch disease. All his MRI and EEG lesions supported disease in that area. And presumably, he was infected with divergence notocolii when he got that rash. Was it a tick? No one ever saw a tick. And so presumably co-infection contributed to his seizures, which were extremely refractory, and his histologic diagnosis that did not fit Rasmussen's encephalitis, so they're not going to cut out half his brain. And with that, I really want to thank the Cohen Foundation for supporting our efforts to improve diagnostics and direct detection of these pathogens. Thank you.
Thanks so much, Ed, and thank you for sharing that case with us. I think it's really important to have specific cases that we can discuss. It helps integrate what we're learning, and we've heard about Lyme, Bartonella. We're going to move to COVID now and hear some common symptoms that we're seeing in long COVID and IACI.
Great, wonderful. Thank you so much, and thank you for having me here and for sharing some of the experiences with our patients who are coming to us to a clinic at Columbia with long COVID symptoms. And really, much of this work is very sort of translatable to experiences for patients with infection-associated chronic illnesses. So what I'm going to share with you is just some guiding principles, how we approach patient care. And much of it is really informed by kind of discussions that you've actually been hearing, we've been hearing all day today, as well as sort of experiences that we've had with patients living with HIV. As we all know, sort of major proposed mechanisms that kind of are underlying infection-associated illnesses and long COVID are actually similar, which makes it easy but also challenging to kind of to find unifying or simple diagnostic tools. Some guiding principles I've listed here on this slide. Because symptomatology spans so many different body systems, we know it's incredibly important to sort of approach this in a multidisciplinary fashion and follow patients longitudinally, because symptoms wax and wane. So if we want to find good biomarkers that may predict risk of progression, we have to collect the data longitudinally when patients are feeling well, and then also when they're feeling worse. And also education of providers and early stage trainees, and incredibly important. We've heard about this earlier in the sessions. So that we can train individuals who are actually sort of facile in diagnosing patients presenting with long COVID and infection-associated chronic conditions, and can kind of tap into multidisciplinary care and collaborate with individuals across different disciplines. And listed here is kind of an approach that's taken by many clinics, and certainly ours in terms of referrals and cross-referrals to physical therapy, cardiologists, neurologists, and mental health specialists. What I've listed here on this slide is just the approach that we are taking to our patients with long COVID and infection-associated chronic illnesses in terms of kind of a thorough workup and diagnostic evaluation. And really, a lot of it starts with history. And a good, well-taken history and documented history is kind of always what anchors our subsequent follow-up, as well as using validated measures to clinical assessments to document presence or absence of dysautonomia, POTS, neurocognitive symptoms. And it's important to track these longitudinally, so these questionnaires we administer actually over time. Sort of exam is equally important, as it's sort of laboratory assessment, looking for pathogens that may have been present or footprints of the pathogens that may have caused kind of current symptoms. We always we've heard a lot about kind of the inflammatory and immune response to these kind of that are present in these conditions. So looking for biomarkers of this in terms of even sort of elevations in cytokines is an important part of the evaluation. And then imaging and advanced testing sort of is guided by the individual's presentation. So we may refer for chest CTs, cardiac evaluation, and neurological evaluation is a really important component of the diagnosis. In closing, because I want to make sure we leave some time for question, I do want to just kind of emphasize how sort of taking this multidisciplinary approach to diagnosis and care of patients who present to us is incredibly important, just to kind of make sure that we have a good longitudinal trajectory and data points as we look at biomarkers of improvement and recovery. Embracing intersectionality, we all talk is sort of is key. I think both kind of in the care as well as diagnosis, and kind of keeping an open mind is incredibly important. And I think sort of approaching this kind of through a very patient-focused approach, and making sure that we include patients' sort of voices about what works and what doesn't work is incredibly important. So I wanted to close with sort of really special thanks to not just our funders, and especially Stephen and Alexandra Cohen Foundation, but to our patients and the providers who really are kind of helping us learn a lot about how to diagnose and then care for patients who present to us. Thank you.
Thanks so much, Magdalena. All right, well, we do have some time for questions. I'd also like to kick it off by just saying I'm really cheering you all on, because really any of the lessons that you learn will probably inform the work of each other, and that is the beauty of having this broader group of infection-associated chronic illness activated and working together. At CDC, certainly we are looking at the long COVID folks. I've got my colleague from MECFS here, so there she is. Certainly within the Lyme program, West Nile, dengue, we have all of these infection-associated chronic illnesses spread out across CDC, just as it's spread out across the field. So really, really cheering you guys on, and hope we find some discoveries around diagnosis for IACI. And with that, I'll take any questions that you might have from the audience. Yes, Sue.
Question for Dr. Breichford. Sorry, with the little boy with the Bartonellosis, was he treated? And if so, did it help resolve the seizures?
Unfortunately, he's never been treated directly for either infection. He has been treated with IVIG for autoimmune encephalitis and did improve with IVIG therapy to a certain extent.
Yes, ma'am.
Thank you. Wow. I have long COVID and a whole bunch of infection-associated chronic conditions. And I'm now co-author research, do a lot of stuff with researchers in government agencies and legislation. And I help patients all over the country get diagnosed, especially patients of color, with all of these different illnesses quite easily, just from a couple of conversations with them. Partly due to my knowledge of dysautonomia, MCAS, and MECFS. And every single patient that I've talked to that gets sick within about 6 to 9 months, none of them have become as severe as I have. So I do believe that there is a path forward if we look at what we already know with the illnesses. Like clockwork, since I'm from the March 2020 crew, we're seeing patients follow the path of MECFS and dysautonomia, developing cancer around the same times, getting resistant to therapies for those who got sick from the later waves, at that 4 to 5 mark that MECFS patients have been saying anecdotally for years. And so my question is, why still is there so much resistance to just embrace what we already know, and especially what patients know about clusters of symptoms, to develop diagnostics and also some predictive modeling so that people like me would know for sure in 10 years what my life's going to look like if I don't get any kind of care. I feel like when I talk to researchers that they still tend to kind of want to go down the traditional pipeline, whereas I think if we step back, listen to the patients about what symptom clusters are there, what we can actually test and measure, and not go so deep into the cellular thing that has an insurmountable amount of variables, and start rolling some simpler diagnostics that way. This is curious.
I'd love to take a stab, because one thing that I actually think is, I think is incredibly important, is there's a tremendous amount of information we try to get from patients about their symptoms. And this is something they can most accurately report out to us. And you saw there's a lot of surveys that we use and validated instruments that rely on those symptoms. Patients are desperate and trying everything they can get access to at home. And they know if they feel better. They can track their own symptoms. That's, I feel like, not what they need from us. I feel like our mission needs to be to provide, to understand the mechanisms of disease and provide measures and diagnostics that are tracking those things. Because I think if we're trying new interventions, there's both symptom management, think Tylenol, obviously not Tylenol, but there's that kind of thing that are gonna have an immediate effect that a person can feel. And there's gonna be drugs like biologics, perhaps, that, an example, the IgE example that I gave, so IgE will lead to mast cell activation, will lead to connective tissue breakdown, will lead to a whole lot of misery. Now, if we can see that we're trying something, let's say we're trying an IgE blocker, or we're trying mast cell stabilizers, or and we can see that we're stopping that way upstream, way before you feel it. And so here is where we can actually provide value that you don't know, you're trying something, maybe you don't feel an effect. Maybe you need a higher dose. Maybe you need to be on it for 2 months to feel an effect. You can't tell us that. We could tell you that if we do the right tests. And that's where I feel like we need to add the value with diagnostics, where we can actually tell you about things that haven't happened yet, but that are, which road you're on.
Yeah, and just to sort of add, so first of all, thank you for your advocacy, and kind of in this field, it's incredibly important to have that, and especially as you said, sort of, and I think this was also sort of mentioned in other groups, and having sort of non-white individuals of color, right, who kind of, and having their voices represented for sure. And I think I would just echo kind of I think it's incredibly important to follow people longitudinally, not only to kind of develop these trusting relationships so people can share their symptomatology, but to be able to collect, right, data as well as biomarkers of that can help us sort of predict kind of these sort of ups and downs in a more kind of, precision-driven fashion.
So I have sort of a follow-up question in terms of promise for diagnosis of IACI. Does the panel here, do you think that there's more promise trying to identify a pathogen from perhaps long ago, or a disease process that resulted from the pathogen when we think about diagnostics for IACI?
I'll be happy to take a shot at that one. I grew up on a farm, and I think in very simple farm practical terms. And as a veterinarian, I see immune-mediated hemolytic anemia, immune-mediated thrombocytopenia, immune-mediated polyarthritis, and we put idiopathic adjacent to about 3/4 of those. And in other instances, we find infectious agents, we treat the infectious agent, and the immune-mediated disease goes away. My bias for many years is if we had tests that were sensitive enough, as I hopefully illustrated in the case that I presented, and we had targeted therapies that could eliminate the pathogens, we could put the immunologists out of business. No, just kidding.
No, I hope so. I'm rooting for you.
So I believe it's the pathogen, and I believe in persistent infections, and I'm hoping as a clinician that it's a finite number of organisms that cause persistent infections. If not, I think those of us that see patients want to stop.
Thanks, Ed. And Chris, did you want to? You said both.
Well, yeah, I mean, I feel I've been blessed by not having a medical background and training, and this means that I am not afflicted by the dogma of being told, well, post-treatment Lyme disease doesn't really exist. And so for me, the question is there a single pathogen? Is it a community result, or is it an immune system responding to both of those? And I think we're going to find that there is a spectrum of these mechanisms. And this was pointed out multiple times by Amy and Dave today, that these are complex conditions. There's not We shouldn't expect that there's a single silver bullet because there isn't a single pathology.
Right. Well, I think we're at time. Thank you all for joining us and for participating in this conversation. And thanks to my panelists here for sharing your brain and your work and your career dedication. Thanks so much.